Good evening. I'm Rick Lifton, the president of the Rockefeller University, and I'm delighted to welcome all of you to the 21st annual presentation of the Pearl Meister Greengard Prize. It's just great to have so many of you here with us in person today, and I'm also pleased to welcome those of you who are joining us by webcast. The Pearl Meister Greengard Prize is a cherished evening here at Rockefeller in which we recognize women scientists who have shaped modern biomedical research. For those of you who might be with us for the first time, I'd like to share a little background about the university and this wonderful prize. The university was founded in 1901 by John D. Rockefeller, who recognized that the major limit to preventing or treating diseases was our lack of understanding of their fundamental causes. The Rockefeller University was created to bridge this gap, embracing the credo, science for the benefit of humanity, which has fueled our efforts for the last 125 years. Some of the most transformational discoveries in the last century of biomedicine have occurred here at Rockefeller, including the discovery that DNA is the chemical of heredity. In our 123-year history, with only 257 full professors in our history, 26 of them have won the Nobel Prize in medicine or chemistry. These recipients have been trailblazers in molecular biology, chemistry, cancer research, immunology, biochemistry, genetics, biophysics, and neuroscience. Among our Nobel laureates is the late Paul Greengard, a pioneering neuroscientist. Paul received the Nobel Prize in the year 2000 in physiology or medicine for his fundamental discoveries about the mechanisms of signal transduction in the brain that regulate the activity of neurons. His work contributed profoundly to our understanding of neurologic and psychiatric diseases, including Parkinson's disease, depression, schizophrenia, and Alzheimer's disease. Paul and his wife, the renowned sculptor Ursula von Riedingsvard, who is here with us this evening, established the Pearl Meister Greengard Prize with his monetary share of the Nobel Award and named it after Paul's mother, Pearl Meister Greengard, who died giving birth to him. Paul and Ursula were guided by their belief that women scientists have not been appropriately recognized for their contributions to science, and they set out to change this. Paul and Ursula's vision and generosity inspired friends of the university to add their own support to this prize, which now includes a $100,000 honorarium. To date, 4 recipients of the Pearl Meister Greengard Prize, Elizabeth Blackburn, Carol Greider, Jennifer Doudna, and Catalin Carrico, have gone on to win the Nobel Prize. Past recipients of this prize are commemorated in our Pearl Meister Greengard Prize exhibit in the Abby Aldrich Rockefeller Reception Hall just outside this hallway. And I invite you all to take a look at it after the ceremony if you didn't get a chance to do that during the reception. I'm pleased to acknowledge my colleagues on the Pearl Meister Greengard Prize Selection Committee. We're fortunate to have a very distinguished group of scientific leaders who take part in the selection process, including 3 Nobel laureates. The complete committee list can be found in tonight's program. This evening, we're recognizing Rockefeller's own Svetlana Mojsov, whose discovery of glucagon-like peptide can't even get the words out. Please hold your applause until the end. Tonight, we're recognizing Rockefeller's own Svetlana Mojsov, whose discovery of glucagon-like peptide 1 called GLP-1 and its critical role in the regulation of insulin secretion and blood glucose. This discovery led to development of a revolutionary new class of medicines, GLP-1 agonists, that lower blood glucose, cause dramatic weight loss, and reduce the risk of heart attacks and strokes. And the world has noticed. The benefits of GLP-1 agonists were lauded by Science magazine as the 2023 Science Breakthrough of the Year, and Svetlana's key role in this story is also now being recognized. She was named by Nature magazine to Nature's 10 as one of the 10 people who shaped global science in 2023. I'm also delighted to welcome to our welcome this evening family members of Svetlana's who are with us in the audience. Joining Svetlana is her husband and fellow Rockefeller scientist, Michelle Nussenzweig, and their daughter, Maya. Thank you for being with us tonight. So it's great to see so many people from Rockefeller here with us tonight to celebrate a member of our community, which we will do shortly. But now I want to turn to a special part of our program. As part of the Pearl Meister Greengard Prize presentation, every year we have a tradition of inviting a distinguished woman from a different field to present the prize. This year we are thrilled to welcome Diane Ackerman, who is a luminary figure in the literary world. From her early days as a naturalist and poet to her emergence as one of the most celebrated nonfiction writers of our time, Diane's career has been extraordinary. Her journey as a writer began early as an imaginative child in Illinois, and as an undergraduate, she intended to study psychology. When a computer error at Penn State University declared her major instead to be English, she went along for the ride. Following graduation, Diane attended Cornell, where she steeped herself in literature, poetry, and psychology, laying the foundation for her future work. She received both a Master of Fine Arts and PhD from Cornell. Among the members of her dissertation committee was the astronomer Carl Sagan, who was reportedly an avid fan of the planets, Diane's book of poems about the solar system. As an author, Diane has penned numerous best-selling books spanning a wide range of subjects, from the wonders of the natural world to the complexities of human emotion. Her works, including The Zookeeper's Wife, A Natural History of the Senses, and most recently, The Human Age, have captivated readers worldwide and earned her critical acclaim. Diane is also an accomplished poet and essayist whose writing has been featured in publications including the New York Times, National Geographic, The New Yorker, and many more. Her work has also traveled to the screen. In 1995, her book, A Natural History of the Senses, an exploration of the physical processes underlying our sensations, inspired a PBS Nova series, which she hosted. And The Zookeeper's Wife, a moving and true story of two zookeepers of the Warsaw Zoo in Poland who hid and saved 300 Jews from the Nazi occupation of Warsaw during World War II, was turned into a major movie starring Jessica Chastain in 2017. Diane's impact extends far beyond her published works. She has committed herself to education with teaching positions at Columbia, Cornell, and NYU. She's also a passionate advocate for environmental conservation and animal welfare, using her platform to raise awareness about pressing issues facing our planet. Describing herself as an earth ecstatic, she reminds her readers that human nature and nature writ large are inextricably linked. For her many contributions, Diane has been honored with numerous awards and accolades, including the Stephen Hawking Medal for Communicating Science, the Henry David Thoreau Prize, and an Orion Book Award. She has also published a memoir titled 100 Names for Love, which was a finalist for the Pulitzer Prize and National Book Circle Critics Award. In a non-literary category, Diane has the unusual distinction of having a biological signaling molecule named after her, Diane Ackerman, which is a sex hormone in crocodilians. It's a unique accomplishment. Congratulations. Diane also has a special connection to Rockefeller, as she has served for many years on the selection committee for our Lewis Thomas Prize for writing about science. We're truly honored to have Diane here with us this evening. Please join me in welcoming her to the stage.
Thank you so much, Rick, for that kind and incredibly generous introduction. It's always a pleasure to be here at Rockefeller among so many lively, innovative minds. And I want to thank the members of the selection committee for the great honor of being asked to participate tonight in this prestigious event celebrating the distinguished scientists Dr. Svetlana Mojsov, for her extraordinary achievements in biomedical research, as well as her inspiring career. I also want to thank the renowned sculptor, Ursula von Reidingsvard, and her husband, the late Paul Greengard, for establishing a prize to recognize the momentous, often life-changing contributions of women in science. I'm especially delighted that the award comes from the shared heart of a couple who bridge the arts and sciences themselves. I've spent some of the happiest hours of my life on that uniquely fascinating Escher-like bridge, even though it makes it nearly impossible to know what to say when I'm asked, "What are your books about?" To make it simple, I usually say I'm a nature writer. And that is true, but only if what we mean by nature is the full sum of creation, from the Big Bang to the whole shebang, from the invisibly distant to the invisibly minute. And such common marvels as owl moths with stunning eyes on their hind wings, whales using rhyme to remember their epic songs, spring moving north at 13 miles a day, the harvest moon rising like slow thunder, mirage-like mosaic octopuses, memories wobbling aspic, the night sky's distant leak of suns, an aging father's voice so husky it could pull a sled, the courtship pantomimes of albatrosses, raindrops shape-shifting 300 times a second as they fall, and of course, the several hundred tardigrades that I keep as fascinating and indomitable pets. In short, nature, homesteading every pore and crevice of Earth with endless variations on such basic biological themes as tree frogs, hairy frogs, frogs with sticky toes, marsupial frogs, toe-tapping frogs, and even transparent glass frogs. I grew up in rural Illinois in the jet-propelled 50s, basking in the novelty of black and white television, but of course minus computers and cell phones. So I mainly played outside, and nothing delighted me more than to be up to my shoulders inside a tree trunk, or watching all the things that squirrels do with their tails. It amazed me then, and it still does, how many marvels are bustling through any slender moment. All the textures of life, all the sensations of being alive. I found nature a limitless source of surprises, spectacles, and wonder. That's where I first felt a sense of belonging to the pervasive mystery of nature, of being finite in the face of the infinite, and where I developed the well springs of a secular eco-spirituality. Yes, and became an earth ecstatic. But I also developed a sense of mission. More than anything, I wanted somehow to portray all of those sensations, all of those physical details, the whole big, blooming, buzzing experience of being alive. By the time I was 14, I was trying to capture small shards and sticky fragments of the world in words by writing poems. And I desperately wanted one day to be a sort of scientist poet who patrolled the wilds of the world and the emotional jungle of ourselves with snares made of sentences and darts tipped in the curare of wonder. I also sometimes fantasized about being part of an itinerant band of extraterrestrial artists who visited different solar systems and were reborn into each one, creating art as one sentient life form after another. Their home planet boasted a library which contained the lamplit sagas, unique senses, and contrapuntal natures of many civilizations and entities. This time around, I just happened to be a human on a sunny blue marble in our curve of space. Then when I was 17, I was paging through the local newspaper when I came upon a tiny article which matter-of-factly reported that scientists had discovered that schizophrenics' sweat smells different from other people's. Well, I was astonished. Was it really possible that mind, personality, mood, and other phantoms could in fact be physical, something in a person's unique broth? I was tantalized by the thought and applied to Boston University to study neuroscience while also writing. But when, for various reasons, I transferred colleges to Penn State as a sophomore, just as Rick said, the computer put me in English by mistake. And since I'd been writing shyly but enthusiastically my whole life, and I wasn't very old, I considered it fate. One afternoon, when I was a senior, I was listening to Gustav Holst's gorgeous musical suite, The Planets. I was enthralled by its lyrical flights, but I was also puzzled by our human need to turn the planet Mars into a war god and Venus into a femme fatale in order to appreciate them. I'd been reading general science books and magazines for fun, and the real planets with their bizarre atmospheres and their mysterious terrains completely enchanted me. Why weren't we able to appreciate them and the rest of nature as it physically is? Why did we have to only be able to mythologize them or domesticate them? So I began planning a book-length suite of poems based on the science of the planets. Alas, this was far from popular when I entered Cornell a few months later as a graduate student in creative writing. One day, the senior literary critic took me aside and said with sincere, avuncular concern, "What's a nice girl like you doing writing about amino acids?" "Science has no heart," he said, "no feelings." Well, I guess he didn't know many scientists. I tried to explain that I didn't think the world was knowable from just one perspective, and that I took the universe literally as one verse. That the whole big, buzzing, blooming spectacle of life fascinated me, and that since we live in a world where neurons, viruses, airfoils, and yes, amino acids were such a common part of our everyday lives, not to write about them would mean perversely ignoring so much of life's wonders and variety. He just shook his head and walked away. I was a hopeless case. But luckily, I was able to put on my MFA and PhD committees A.R. Ammons, a poet whose background was in glass chemistry, and Carl Sagan, astronomer who loved the arts. They ran interference for me and made it possible to pursue an interdisciplinary MFA and PhD, technically in English, while poaching in the sciences. Thanks to Carl, I was also able to pore over NASA photos and maps, attend planetary flybys, and enter the Journalist in Space project, which, of course, was canceled after the Challenger tragedy. And by the way, I was among the semifinalists in that. All of the men were photographed looking forward, looking strong. All of the women were asked to pose in a kind of high school yearbook way. That's how our photos were taken. An especially fascinating and fun course that I taught at Cornell was on creativity in the arts and sciences. It was a class full of academic misfits like me. I remember one student who wished to join the class saying, "I'm in high-energy physics, but my real passion is for medieval Irish song." I said, "Great, you're in." I also began joining expeditions then to work with endangered animals, trying to contribute in some small way to their survival, hands-on, but also by writing about them, mainly for The New Yorker and National Geographic and in several books. Much has happened in my writerly life since then. I continue to write books of poetry and poetic prose, and I feel so grateful to have a calling that allows me to create my own astonishment. Writing is also the way that I keep learning about the world because, I admit it, I just love being a student. I'd hate to give that up. And that may inspire me to write about the mystery and marvel of the brain, or the Anthropocene, or A Natural History of Love, or The Dark Night of the Soul, or the compassionate heroism of Antonina Żabinska, the zookeeper's wife, during World War II, or the book I'm working on now about how much our sensory lives have changed since I published A Natural History of the Senses in 1990. Since then, in all of my books, I like to focus on a facet of nature we view too narrowly and try to demystify it, clarify it, give it a human face, imagine it in new ways, celebrate its richness and variety. What I find most fascinating is writing at that crossroads where nature and human nature meet and can shed light on each other. But my books are all part of the same lifelong quest to somehow make the world sayable and to better understand what it was like to have once been alive on this planet. How did it feel in our senses, passions, and contemplations? What did it taste like, look like, smell like, hurt like, yearn like, thrill like? In retrospect, it seems like everything I've written thus far is part of a single mosaic that I could never finish, but which, if I could, would help me better understand the complex saga of human experience. Deep down, I know that that should take from birth to death, at least, and include all of consciousness. So I suppose I should find that overwhelming. But I just don't picture it in that way in terms of success or goal, but rather as a simple mystery trip. The world revealing itself, human nature revealing itself, are so seductive and startling, and that's always been fascinating enough to send words down my spine. Thank you.
Thank you, Diane, for those beautiful words. So, turning to tonight's honoree, we are thrilled to honor Rockefeller's own Svetlana Mojsov with the Pearl Meister Greengard Prize. Svetlana's discovery of GLP-1 laid the groundwork for the revolutionary class of medicines for diabetes and obesity that have been so much in the news in the last year. These amazing medicines lower blood glucose, reduce obesity, reduce risk of heart attacks and death in people with established heart disease, and are currently under study for many other complications of obesity and diabetes, as well as a wide range of other diseases, ranging from Parkinson's disease to addictive behavior. As an independent investigator at Massachusetts General Hospital in Boston in the 1980s, Svetlana discovered a novel intestinal hormone, a short peptide called GLP-1, that's released in response to food ingestion and primes the pancreas to increase insulin secretion. Svetlana came to this project with deep expertise in protein biochemistry and the synthesis and purification of peptides. This left her uniquely positioned to solve questions surrounding GLP-1. For years, however, her indispensable contributions were overlooked and have only recently begun receiving their much-deserved recognition. Svetlana was born in Skopje, the former Yugoslavia. Growing up, she collected butterflies and beetles and kept them in boxes. Attending middle school in Belgrade, she was captivated by physics and chemistry in science and lab classes. Svetlana went on to university and got her undergraduate degree in physical chemistry from Belgrade University. And in 1972, she came to the United States in the PhD program here at Rockefeller, and she joined the laboratory of biochemistry professor Bruce Merrifield. Merrifield had devised a faster, simpler way to synthesize proteins and peptides. His method, known as solid phase synthesis, dramatically increased the yield of desired proteins while reducing the time and cost of their synthesis. This advance earned him the Nobel Prize in Chemistry in 1984. For her dissertation research, Svetlana aimed to use solid phase techniques to synthesize a peptide hormone called glucagon, which increases blood glucose levels. This, however, was widely considered an impossible mission because other labs had already tried to do it and failed. But Svetlana courageously persevered, employing an alternate solid phase protocol, and after much painstaking work, she succeeded. She received her PhD in 1978 and continued in the Merrifield lab as a postdoc and research associate for the next 5 years. During this time, she married Michel Nussenzweig, who was a classmate who earned his PhD in immunology at Rockefeller while also studying medicine at New York University. In 1993, they moved to Boston, where he started his residency training at Massachusetts General Hospital. Svetlana was hired into the MGH endocrine unit as director of the Howard Hughes Medical Institute-supported peptide synthesis facility, and she was also appointed an instructor in medicine at Harvard Medical School. Her position afforded her the opportunity to pursue her own research. A veteran of glucagon biology and chemistry, when the gene encoding glucagon from mammalian species, including humans, was published in the summer of 1983, it immediately got her attention. The gene encoded a protein of 180 amino acids, but a string of only 29 of those amino acids comprised the active hormone glucagon, which was embedded in the larger precursor and was later clipped out. Inspection of the rest of the amino acid sequence of the longer precursor protein revealed that it contained several potential additional small peptides that were entirely unknown at that time, but could be other hormones. These were called GLP-1 and GLP-2, but precisely where they started and where they ended was completely unknown. It had been speculated by others that a peptide embedded in the precursor protein might be the long-sought incretin hormone, a hormone that's made by intestines that primes the pancreas to secrete insulin after a meal. This hormone had first been suggested in the first part of the 20th century, but had never been identified. Svetlana trained her attention on GLP-1. Others who had studied the sequence of the precursor protein thought that the active form of GLP-1 would be a chain of 37 amino acids clipped out from the precursor. However, in perusing the sequence, Svetlana immediately predicted a truncated structure for GLP-1 that was clipped out at another site, producing a peptide of only 31 amino acids. This insight proved to make all the difference. In September of 1983, Svetlana began independent work testing her hypothesis. She used her skill to synthesize the longer and shorter proposed structures for GLP-1. She made antibodies to the peptides and found evidence that GLP-1 was indeed made in the intestines. Collaborative work with Joel Habner's lab at MGH also found evidence that the precursor protein was expressed in the intestine. These findings, published in a pivotal paper in 1986, supported GLP-1's potential role as the long-sought incretin hormone. Other collaborative studies followed with Gordon Weir and David Nathan, all using the alternative forms of GLP-1 that Svetlana had synthesized. The work proved that Svetlana's 31 amino acid form of GLP-1, but not the longer version, potently stimulated insulin secretion from the rat pancreas. And in further work in 1982, the first study in humans was performed in collaboration with David Nathan at MGH, showing that her GLP-1 lowered blood sugar levels in diabetic and non-diabetic individuals. By that time, Mojsov had relocated to Manhattan, where where she and her husband had a young son and a baby daughter. Michelle had landed a job here at Rockefeller and was a rising star in immunology. Svetlana joined the Rockefeller lab of immunology pioneer and future Nobel laureate Ralph Steinman, where she became a research associate professor. And with her own funding, she has continued to investigate GLP-1 and glucagon biology in mammals and fish, taking an evolutionary perspective to the problem. And that research continues to today. So efforts to turn GLP-1 into a useful drug was immediately an attractive idea, but it had many challenges. Specifically, it was only made at very low levels and was rapidly degraded when it was injected. The pharmaceutical company Novo Nordisk produced the drug Victoza, a version of GLP-1 that modestly improved blood sugar control, but nonetheless was a useful drug in the treatment of diabetes that was approved by the FDA in 2010. More successfully, Merck found that they could sustain low levels of GLP-1 by inhibiting the enzyme that's responsible for GLP-1's degradation. This resulted in the medicine Januvia that produces glucose-dependent insulin secretion, which lowers blood glucose in diabetics, but because its action depends upon the presence of glucose, dramatically cuts the risk of dangerous low blood sugar that complicates the use of many other drugs used to treat type 2 diabetes. Januvia became a mainstay of the treatment of type 2 diabetes following its approval in 2006. But Novo Nordisk, in the background, continued efforts for the next decade to prolong the half-life of GLP-1. And after 10 years of work, they succeeded, principally by adding a fatty acid moiety to the molecule and eliminating the cleavage site for DPP-4 and chopping off the amino terminal part of GLP-1. This allowed high levels of this version of GLP-1, now marketed as Ozempic. When used in clinical trials as a self-administered once-weekly subcutaneous injection, it produced improved control of blood glucose, and the FDA approved its use in 2017. But these trials also revealed a surprising effect. These higher levels of GLP-1 were producing weight loss in obese patients. Further studies showed that still higher doses produced truly remarkable weight loss, averaging 15% of body weight in obese or overweight individuals. Moreover, after 3 years of treatment, weight loss was sustained, and patients with pre-existing cardiovascular disease had a 20% reduction in heart attacks, strokes, cardiovascular death, and all-cause death. This higher dose, now marketed as Wegovy, received FDA approval in March of 2023. A related drug, Mounjaro from Lilly, has similar effects and was approved in November. The response to these drugs has been nothing short of astonishing. In the year 2023, starting from a very low level of usage, close to 1% of the U.S. population has been prescribed Ozempic or Wegovy. In sum, Svetlana's seminal contributions have already benefited millions of people. There is nonetheless another notable aspect to this story that's germane to tonight's prize. Novo Nordisk started work in this field by licensing patents from Massachusetts General Hospital, which had secured these patents based on Svetlana's discovery of the biologically active GLP-1 peptide. However, MGH filed these patents without informing or crediting Svetlana. She only became aware of this oversight a decade later and fought for the next 10 years to be rightfully included on the 4 issued patents related to the discovery and use of GLP-1. The U.S. Patent Office ultimately issued the corrections to these 4 patents. Recently, prestigious scientific prizes, including the Gairdner International Award, were awarded for GLP-1's discovery without acknowledging Svetlana's seminal role. When her name was mentioned at all, she was listed as a technician in Joel Habener's laboratory, which was never the case. But science journalists reporting the story uncovered the true history of GLP-1's discovery, which resulted last fall in high-profile reports in Nature, Science, and the biotechnology journal Stat. Since then, the tide has begun to shift. In December, Svetlana was recognized as a co-recipient of the 2023 Vin Future Prize, established by a Vietnamese philanthropist for the discovery and use of GLP-1. In December, she was also named to the Nature 10 list of Nature magazine's list of the 10 people who most helped shape science in 2023. The Pearl Meister Greengard Prize was established specifically to recognize women in science because their contributions were too often ignored or overlooked. While this prize has not previously been given to a member of the Rockefeller community, the prize jury felt strongly that Svetlana is the perfect example of why this prize was created and has been necessary. I strongly believe that Paul Greengard would be extremely proud that this year's award is recognizing Svetlana. And I know that Ursula strongly agrees with this sentiment. And as if on cue, just today, Time magazine released their Time 100 list of the most influential people worldwide for 2024, and this list includes Svetlana Mojsov. I have no doubt that other important prizes and awards will follow. Svetlana never fails to emphasize her good fortune in having had excellent scientific mentors and collaborators in Bruce Merrifield and Ralph Steinman. She is equally proud of the young scientists that she has mentored during her own career, and I am truly touched to note that Svetlana will donate her Pearl Meister Greengard Prize money to Rockefeller's Women in Science initiative to support the careers of graduate and postdoctoral scientists at Rockefeller. We're so incredibly proud to pay tribute to Svetlana and her foundational scientific accomplishments with the Pearl Meister Greengard Prize. Please join me now in welcoming her to the stage. Diane, come up here.
This way?
So you here. So wonderful. So we'll take photos afterwards. Congratulations. So I'll now read from the citation that is being presented to Svetlana this evening. Svetlana Mojsov, you are being honored for your profound insight, brilliant biochemistry, and critical experimentation that resulted in the discovery of GLP-1. You identified the biologically active peptide, used your biochemical skill to synthesize large quantities of it, and showed that it was made in the intestines, making it likely to be the long-sought incretin hormone that's made in response to food ingestion, preparing the pancreas to increase insulin secretion. You showed that GLP-1 peptide indeed increased insulin secretion when infused into the rat pancreas, and then used it to demonstrate glucose lowering in humans, providing a path to glucose-dependent insulin secretion, safely lowering blood glucose without the risk of hypoglycemia. The resulting medicines transformed the treatment of diabetes. More recently, the ability to increase GLP-1 to much higher levels in the blood has resulted in another therapeutic revolution, with medicines including Ozempic, Wegovy, and Mounjaro that not only substantially lower blood glucose, but also produce remarkable weight loss in obese individuals, reducing the risk of heart attacks and likely a number of other diabetes-associated diseases. Today, in presenting you with the 2024 Pearl Meister Greengard Prize, we recognize and thank you for your seminal work discovering and revealing the physiologic function of GLP-1, paving the way to novel therapies that have improved the lives of millions of people. And Diane will now present the award.
It is my great honor to present to Dr. Svetlana Mojsov the 2024 Pearl Meister Greengard Prize, an international award recognizing outstanding women in biomedical science. On behalf of the Distinguished Selection Committee and all of us here today, we congratulate you heartily on this award.
Well, Rick, thank you very much. This was the most generous introduction I could possibly get, and most generous description of my work. Thank you. And thank you, Diane. I am very honored to receive the Pearl Meister Greengard Award and want to thank the members of the selection committee for including me in the company of women scientists who made major contributions to the advancement of our scientific knowledge. I am especially happy that my discovery has impacted the lives of so many people. This recognition will always have a special meaning for me, as I am receiving it from my alma mater, where I grew up scientifically as a graduate student in Bruce Merrifield Group. When I returned in 1990 to Rockefeller University, Ralph Steinman's laboratory became my scientific home. Both Bruce Merrifield and Ralph Steinman were inspiring scientists and inspiring mentors and big supporters of women in science. Their laboratory were a melting pot for a generation of young people who came from different cultural backgrounds and shared a common desire to learn science and do science. Lab members established long-lasting collaborations and strong friendships. I am deeply touched, really, by the warm letters of support I received from my colleagues from Merrifield and Steinman Group and from my classmates from graduate school. Above all, I want to thank the entire Rockefeller community for their support. From Rick Lifton, thank you, Rick, and especially Mike Young, to the members of the faculty, the administration, and the staff. Because Rockefeller has been such a special place for me, I have decided, as Rick Lifton already mentioned, to donate the prize funds to Rockefeller University and the Women in Science Initiative. Thank you all. Sure. I may take some water.
Give you a moment.
Yes. Yes, thanks.
Take your time.
All right.
We've got nowhere else to go.
Okay, we can start.
Okay. So let's go back to the beginning. So you were born and raised in Yugoslavia, and I understand that education there at the time had quite an egalitarian spirit, where gender didn't seem to make much difference in terms of what you might choose to pursue. If that's true, I wonder what it was like for you coming from Yugoslavia to the United States and entering a graduate program in biochemistry.
Well, Rockefeller was a great place. I was asked that question during my actual interview at Rockefeller University by the dean at the time, Dr. Frank Brink. And I was taken back. I told him that I never knew that there were men's and women's profession. I sometimes think that's how I was accepted at Rockefeller. But I was never asked that question anymore. I think Rockefeller was a very supportive environment, and we had great classmates, and we never discussed that.
That's encouraging to hear. So after you spent all your time learning solid phase biochemistry and peptide synthesis, went off to Mass General, and it seems like it was perfect crystallization of opportunity to be running the peptide synthesis unit with HHMI support with a level of independence to work on what interested you. That must have been a special environment to be in.
Well, yes, the endocrine unit was the perfect environment. They were very collaborative and very supportive. And we had similar interests. They use solid phase synthesis. And that's how that's how recruited me to come there. And they were studying the biochemistry of the parathyroid hormone, which really went long way. And so and Howard Hughes was very generous, and it allowed me really to have the freedom to explore my own interests. So when the glucagon gene was published, it was just as I joined the Mass General. I came in May, and there was first paper already in May, and then in July, and it moved on from there. Actually, I started in September already to synthesize the peptides for making antibodies.
So the key to your success was others had looked at the sequence and made deductions as to what the active form of the likely peptide would be. But you made a very different deduction from what every other group that I'm aware of had made at the time. What was it that gave you that insight that others missed?
So it was actually my training in peptide synthesis. So let's see, and in my knowledge of glucagon. And so when I compared the synthesis of the sequence of GLP-1 for that of glucagon, if you look like this, if this is GLP-1 and this is glucagon, what I noticed that actually in the GLP-1 sequence, which was predicted by Bell, there was an amino acid, basic amino acid, arginine, and I realized that's actually a site which is a signal for cleavage by enzymes. And so if you cleave that arginine, it suddenly moved here. And it was perfect alignment. And especially in the parts of the amino acids which were known to be important for biological activity. So I immediately just deduced that will be the biologically active peptide. And moreover, it was known already that even glucagon can stimulate insulin secretion, but it was at very high concentrations. It was not physiological. And that's when it clicked. Oh, so this is the incretin. This is the peptide which is going to stimulate insulin secretion, and it will be intestinal. And then the challenge was really to develop methods which will allow me to fish it out from really the large number of other intestinal peptides. But I had a very good research assistant. She was very dedicated, and the two of us worked on the bench side by side. And within 2 years, we found it. It was in the summer of 1985. So it was kind of very rewarding. I want to mention one thing. Actually, it was Bruce Merrifield who sent me the manual shaker to make GLP-1. Because there was one instrument in the endocrine unit, but it was reserved for the parathyroid hormone peptides. And so I called, there was nothing at that time commercially available. The shakers were made at Rockefeller University by our machine shop. So I called Dr. Merrifield, and I told him what I want to do. And I got the shaker in the mail. And he gave me a very good advice. He said, just make sure you have a hard-covered notebook with dates. You will need it one day. And that helped.
You can see where that played a role years later. It was a very, very good idea at the time. I suspect that, Rockefeller ought to be getting a cut from those patents as well, shouldn't it? So this is great. So the effect on priming the pancreas to make insulin was somewhat anticipated. Was anybody at that time thinking about weight loss as a potential phenotype?
Oh, no. Nobody, and I didn't think of it. Well, this was 1980s, and there was no basis that there was any hormonal basis for obesity. It was actually only after Jeff Friedman discovered leptin here at Rockefeller University that we learned that hormones can have effect on weight loss. Now, for GLP-1, it was first noticed that its effects on weight during the very large clinical study which was done in Germany in the mid-1990s. And every participant lost weight. And I think that was the beginning when Novo Nordisk also started their research to find a stable analog of GLP-1 to be used for weight loss.
One of the big mysteries in the field is how is GLP-1 at pharmacologic doses having these profound effects? What are your thoughts about how this will be explained in the long run?
Well, here we have, so the discovery of GLP-1 was based on the biology. We know what we were looking for. The effects on weight loss were all by clinical use, and so we now have to understand the biology behind that. We know that there are GLP-1 receptors in the brain. Actually, I've shown here in Ralph's lab in the mid-1990s that there was a receptor both there and in the heart. But now that's the next frontier. We have to learn what is the mechanism. We can guess some better that has to do that the signaling of the receptor somehow manages to get into other networks. And that's how it works. But that's the next frontier. We have to learn that.
Yeah, it's quite amazing that already it's clear that the effects on cardiovascular risk are very significant, and you can see that at least large parts of that effect are through traditional cardiovascular risk factors, and it doesn't seem hard to imagine that other complications of diabetes, fatty liver, kidney disease, will be ameliorated by these drugs as well. So you can imagine so many implications for health that are going to come from this amazing result. I mentioned the 1% of the population that's already received a prescription for one of these drugs, and it'll be very hard not to imagine a lot more. The current projections of the use of this will make this, easily one of the most important medicines in humans in all of history.
I just want to add that there is already a second generation of GLP analogs, which are even more effective in reducing weight than Ozempic. This is, again, a peptide, which is both an agonist of GLP-1 and also agonist of a second intestinal peptide, which is gastric inhibitory polypeptide, or GIP. It was actually developed by another alumni of Merrifield Lab, Richard DiMarchi, together with Matthias Chopp. And that's what's now being distributed under the name of Mounjaro and Zepbound.
Yeah, so this is another fascinating drug marketed by Eli Lilly that has even larger effects on weight loss, and people suspect that this may eventually overtake sales of Ozempic. Yeah.
Actually, individuals taking Zepbound now lose up to 25% of their body weight. So I think I'm very optimistic that we will actually find an effective drug for weight loss.
Do you think side effects of these drugs are going to be a problem?
Well, there are some side effects, yes. They're mostly intestinal. But it seems that some people who don't tolerate the Ozempic, there was a recent report that actually Zepbound is much better. And I think that was the rationale which Richard DiMarchi used by adding the gastric inhibitory polypeptide, the GIP, that actually can somehow help, because that actually has effects on the intestine which can help with the other effects from GLP-1.
So your work really was focused on the basic science of what GLP-1 does. I think in the public sphere, we usually look at the drugs and what they do in people and don't pay much attention to where the discoveries actually came from. And this seems to be a current risk in the public discourse, with the NIH budget now increasingly focusing on translational medicine and clinical applications, and not paying enough attention to basic science, because these are where the great ideas for translation are going to come from. What are your thoughts about basic science and its relationship to clinical medicine? You've worked across these fields.
But we know that all the major medicines came from, collaboration between the bench scientists, the chemists, and working with the biologists and clinicians. So I think bench science is critical, and hopefully NIH will realize that and get more funding. I must say, for me, it was very beneficial that I had a Howard Hughes support, because the budget was very generous, and I didn't have to apply for NIH funding. It was not really until I described NIH that I actually did receive an NIH 5-year NIH funding. So we have to keep on pushing that.
Yeah, no, these are the challenges. We have time for some questions from the audience, so if you have a question for Svetlana, please raise your hand, and we have microphones that'll come to you so you can ask your question. So, there's a question over here. Microphone be right with you.
Svetlana, congratulations. And when coming here, I didn't fail to notice the stench of cannabis in the air when walking the sidewalks of Manhattan. And there was recently a report that users of Wegovy, who inject GLP-1 agonists, are protected against cannabis use disorder. So that plays into what Rick mentioned, the other diseases that are out there that are not cardiovascular, that affect human behavior, which are also affected by GLP-1RA. And if you had a say, how would you prioritize? How should we do research and drill into that human brain signaling circuit function, circuit malfunction that is modulated by GLP-1RA medicines? How would you prioritize that? What should we go after in the first place?
Yeah, I think it's as important to do that as looking for weight loss. There are a lot of scientists who will be interested in that. So I think it should be done in parallel. And also we have to understand, I think, from animal studies first, how the signaling to the GLP-1 receptor works. But that's the basic science now. So everything which we see, it came from clinical use, and now we have to understand how the biology works. So that is a priority, but as good as weight loss and treating obesity, that's a major issue now in the Western countries. So they should go in parallel.
Thank you. We have Mary Beth has a question here.
I think I can speak now.
People online won't be able to hear you. Well, you might try that. I told that, right?
First and foremost, congratulations. Story and brilliant insight. So I, of course, am interested in the GLP-1 binding in the brain. And I have a brother who is seriously affected by Parkinson's, which is a tragic disease. And I read last week in Lancet that there have been some promising treatments with GLP-1 drugs which are available. And I wondered if you have any thoughts about that, or whether the work in the brain is, getting going, or what's happening.
Well, I think one of the hypotheses there is that actually GLP-1 may suppress inflammation, and that inflammation may be part of the development of Parkinson's and also the Alzheimer's. So I think the focus should be on that, whether we can understand that. I'm not sure whether there are good animal models for that, but if we can find a way to prove that, again, Not because this is still just small number, right, of maybe that paper, it was maybe about 100 pages. So we have to follow up. And again, it has to be the clinical observation back to the basic science.
Elaine.
Thank you. Let me echo the congratulations. It's wonderful. Just curious about the fact that you had mentioned that the GLP-1 analogs get down the weight by about 15%, maybe now it's 25%, but it's not more than that. And I wonder, is there some kind of feedback mechanism? Do you know anything about why you don't go further, why weight loss doesn't continue to a chronic?
I think there is a feedback mechanism, but it has to be some kind of coordination between the effects in the brain and the signals which will go back to the adipose tissue. And here is where we can maybe go back to leptin and see how whether adding leptin to this or some other peptides in combination with this GLP-1 agonist may help maintain that weight or have it even more sustained.
Thank you. I think we're at time. So once again, Svetlana, congratulations and thank you for all your amazing work. Really wonderful. And lastly, I just want to thank everyone for coming tonight. It's been a very special event for all of us here at Rockefeller. So for everyone in the auditorium and on the webinar, thank you for attending, and I wish you all a good evening.